Now we can see about the next unit, that is unit number 4. It is basics of phytochemistry, and in this unit you have to study about the modern methods of extraction, then application of latest techniques like spectroscopy, then chromatography and electrophoresis in the isolation, purification as well as identification of crude drugs. So that is what you are going to study under this unit. It's somewhat a bigger unit, and here in extraction technique you have to study about different type of extractions, then also about spectroscopy, about the various spectral methods, then regarding chromatography you have to study about all the chromatographic techniques, then electrophoresis, etc. So these are the things what you are going to study under this topic, and you can expect a lot of questions from this unit, maybe for essay question as well as short answers, then even 2 mark question can also be expected from this topic. Okay, so before entering into the topic, first we can see what are the things that you are going to study this unit. Okay, so first thing is about introduction to phytochemistry. So here you have to understand what is phytochemistry, what are the basics of phytochemistry, etc. Some scrub things are there that you have to study regarding this section. Okay, second thing is about extraction and different extraction techniques, and here you have to get an idea regarding what you mean by extraction. So extraction what do you mean by extraction and different extraction techniques. So in your syllabus only it is mentioned like modern extraction techniques. Then also you have to study about all the conventional as well as modern extraction techniques. So it is a big section, and maybe you will be getting an extra essay question from this topic, extraction. So there you have to study about different types of extractions like infusion, decoction, maceration, then percolation, continuous hot oxalate extraction, supercritical fluid extraction, then set box techniques, etc. So these are the different types of extraction techniques that you are going to study under this unit. Means this topic, that is extraction and different extraction techniques. Then third thing is about the various spectroscopic techniques. So I think you already get an idea regarding this from your analysis classes also. So here I have to study about the different spectroscopic methods like UV visible spectroscopy, then IR spectroscopy, infrared means NMR spectroscopy, mass spectroscopy, then fluorimetry, etc. So XRD, etc. So different spectral techniques are there. So അതൊക്കെയാണ് ഇതിനകത്ത് പഠിക്കാനുള്ളത്. Then the fourth thing is about the various chromatographic techniques like paper chromatography, thin layer chromatography, then column chromatography, HPLC, HPTLC, etc. And last thing is about electrophoresis. So I think regarding this electrophoresis, maybe you studied in your inorganic chemistry or some other topics. Okay, so these are the things that you are going to study under this unit. It is a very big unit, and you will be getting a lot of questions. So here you can expect an essay question from extraction, and also you can expect essay question from chromatography, then 5-mark question from extraction, then chromatography, electrophoresis, etc. So and also inside this maybe some 2-mark questions will also ask. Okay, so today you can see about spectral. So extraction first we can see about spectroscopy. So regarding spectroscopy, you have to study about few things. So first you have to understand what is spectroscopy, then what are the different types of spectroscopy available here, and a brief idea about each and every type of spectroscopy. So suppose a question will be asking like this: explain about the various spectral techniques involved in the isolation of natural, means isolation as well as estimation of natural products. So if it is a 5-mark question, then you have to write the entire thing which I am taking here in this lecture along with other one lecture also. So here I will be taking only the half of the topic, and remaining half you can see in the next lecture. Clear? So first we can see about spectroscopy. First you have to understand why we are going for spectroscopy. Clear? So what is the purpose of doing this spectroscopy? First you have to understand that. So here for identifying a plant constituent, you are already knowing that plants were having lot of chemical constituents, and in fact in short we can say that a plant is a biochemical laboratory, and there are lot of chemical components were synthesized. And here for identifying a plant constituent, first we have to do extraction, then it should be isolated, and after isolation that should be purified. Clear? So first it should be isolated, then purified, then the next step is determine the class of compound. Clear? So for identifying a plant constituent, first it is to be isolated, then purified, then determine the class of compound. Clear? So ആ ഒരു സീക്വൻസിൽ തന്നെ നമ്മൾ ഇതങ്ങ് പഠിക്കണം. So for identify means here you have to get an idea why we are doing this spectroscopy. So for that purpose only we are explaining this thing. Okay. So for identifying a plant constituent, first it should be isolated, and after isolation it should be purified. Then you have to determine the class of compound. So here a class of compound എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ you have to identify whether it is alkaloid, then glycoside, then tannin, flavonoid, likewise which class of compound it is. So this you can identify with the help of various chemical tests only. Okay. So after identifying the class of compound, you have to find out which particular substance is that within that class. Clear? So you know a class. So you identify that the given plant is having alkaloids as a constituent. Then the next step is you have to find out which particular alkaloid is present there. അതായത് which particular substance that is within that class. So that you have to identify. Clear? So before doing any test, first you have to confirm the homogeneity of the sample. So homogeneity means you have to clarify that the given sample or means the sample which you are taking is purified or pure in the pure form. That is its homogeneity is to be tested, and this homogeneity can be tested with the help of chromatography. And if in the chromatography, if you are getting a single spot, then you can confirm that that's a purified compound. Then you can go for the above mentioned steps. Clear? So here, first for identifying a plant constituent, it should be isolated, then purified. Then the next step is determine the class of compound. Here class of compound means whether it is alkaloid, glycoside, tannin, flavonoid, etc. And after identifying the class of compounds, before that, how you will be identifying this class of compound? It can be done with the help of lot of chemical tests. So after identifying the class of compound, next step is to find out which particular substance in that class. ആ ക്ലാസ്സിലുള്ള ഏത് സാധനമാണ് അതെന്നുള്ള കാര്യം കണ്ടുപിടിക്കുക. Clear? So before doing all these tests, first you have to confirm the homogeneity of the sample. That is, you have to ensure that the taken sample is pure only. And this purity can be assessed with the help of chromatography. And if in the chromatography, if you are getting a single spot, then you can confirm that the given sample is pure. Clear? So thereby you can confirm the homogeneity of the sample. Clear? So by the help of various spectral techniques, it is here only we are using the spectral techniques, and that is with the help of various spectral techniques, lot of informations regarding the chemical components present in the plant can be identified or noted. Lot of informations can be revealed from the plant constituent, and that can be achieved with the help of different spectral techniques. Clear? Now you have to know about the different spectral methods that are used in the identification of plant constituents. So lot of spectral techniques are there. The various spectral techniques that are used in the identification of plant constituents are, so first one is UV visible spectroscopy. UV spectroscopy as well as visible spectroscopy. Both were working under the same principle only, but a slight difference is there. So that you will be studying later. So the different spectroscopic techniques that are used in the identification of plant constituent. The first one is UV and visible spectroscopy. Second thing is IR spectroscopy or the infrared spectroscopy. And the third thing is NMR spectroscopy or nucleomagnetic resonance spectroscopy. Now the next one is mass spectroscopy or MS. The next one is XRD or X-ray diffraction crystallography. So it can also be considered under this topic only. And the last one is fluorimetry. So these are the different spectral techniques that were employed in the identification of phytoconstituents. Clear? So ഇപ്പോൾ നമ്മൾ ഇത്രയും പഠിച്ച എന്തിനാണ്, എന്തുകൊണ്ട് spectral techniques നമ്മൾ യൂസ് ചെയ്യുന്നു അതിനു വേണ്ടിയാണ്. So here for identifying a plant constituent, so first you have to isolate, then it should be purified, then determine the class of compound, then find out which particular substance is that one in that class. And all these information can be obtained by the help of various spectral characters. Clear? And the different spectroscopic techniques that are used in the identification of phytoconstituents are UV spectroscopy, visible spectroscopy, then IR spectroscopy, NMR spectroscopy, then mass spectroscopy, XRD or X-ray diffraction crystallography, and finally the last one is fluorimetry. So these are the different spectroscopic techniques that were employed in the identification of phytoconstituents. Clear? We can see about the first type of spectroscopy, that is UV spectroscopy. So it is known as ultraviolet spectroscopy. UV spectroscopy is also known as ultraviolet spectroscopy, and here the absorbance spectra of a plant constituent. So in UV spectroscopy, the absorbance spectra of a plant constituent can be measured in very dilute solution. So the absorbance spectra of a plant constituent can be measured in very dilute solutions against a solvent blank with the help of an instrument, and that is known as automatic UV spectrophotometer. Clear? So in UV spectroscopy, the absorbance spectra of a plant constituent can be measured in very dilute solution against a solvent blank with the help of automatic UV visible spectrophotometer. Clear? And here in UV spectroscopy, only colorless compounds can be selected for analysis. So for the identification or for the analytical purpose, only colorless compounds can be used for estimation with the help of UV spectroscopy. Clear? And the wave range in UV spectroscopy is roughly between 200 and 400 nanometer. So ഇത് കുറച്ചുകൂടെ ക്ലിയർ ആയിട്ട് നിങ്ങൾ അനാലിസിസിൽ പഠിക്കും. So 180 to something എന്നുള്ള കാര്യം പഠിക്കും. So but roughly the wave range in UV spectroscopy is from 200 to 400 nanometer. So അപ്പം എന്തൊക്കെയാണ്? In UV visible spectroscopy, the absorbance spectra of a plant constituent can be measured in very dilute solution against a solvent blank, and the instrument user is automatic spectrophotometer. And here only colorless compounds can be taken for analysis, and the rough wave range of UV spectroscopy is between 200 to 400 nanometer. Clear? And with the help of UV spectroscopy, two things can be noted. First one is lambda max, and it is the wavelength at which the maximum absorbance takes place. So one is lambda max, and lambda max is the wavelength at which the maximum absorbance takes place. And second thing is intensity of absorption or the optical density. So these are the two things which we are going to study under this UV spectroscopy. So one is lambda max, it is a wavelength at which the maximum absorbance takes place, and second one is the intensity of absorption or the optical density. Clear? So these are the two things which we are going to study under UV spectroscopy. Clear? So appo ithrayum karyangal clear ayallo. Okay, now both UV as well as visible spectroscopy requires only trace quantity of sample for measurement. That is, it requires only very few amount of sample for the analysis. So this plays a vital role in the analysis of phytoconstituents. Clear? So in UV spectroscopy, the absorbance spectra of a plant constituent can be measured in very dilute solution against a solvent blank, and only colorless compounds can be taken for analysis. Then the next point is the wave range is from 200 to 400 nanometer, and two parameters can be studied. So first one is lambda max, and second one is optical density. Then last point is it requires only trace quantity of sample for the measurement. Clear? So that is about UV spectroscopy. Now we can see about the working principle of UV spectroscopy. See this graph, means graphical diagram. So here UV light should be passed through a colorless substance. I already told that here only colorless substances can be taken for analysis. So you have to pass UV light through a colorless substance, and when light is passed through a colorless substance, so here absorption will be taking place. Absorption will be taking place, and this time measure the intensity of absorption. Measure the intensity of absorption, then note the lambda max. Okay. So intensity means here optical density or the intensity of absorption will be noted, and also note the lambda max, and this can be taken for either quantification or qualification. That is quantitative or qualitative evaluation of the sample, and this is the working principle of UV spectroscopy. So UV light should be passed through a colorless substance, then absorption will be taking place there. This time measure the intensity of absorption, that is optical density should be noted, and also note the lambda max, and these two parameters can be taken for quantitative as well as qualitative evaluation of crude drugs, and this is the working principle of UV spectroscopy. Clear? Now in the last part we can see about the applications of UV spectroscopy. So the first application is it is used in the identification of plant constituent. Roughly proper identify cheyanannu pattilla, so it can be used in the identification of plant constituents. So here the common examples are anthraquinone glycosides will be showing 3 to 4 indents peak between the wave range of 220 and 290 nanometers. So here the first example is identification of plant constituent. അതിനകത്തുള്ള ഡീറ്റെയിൽ ആയിട്ടുള്ള ഒരു കാര്യമാണ് നമ്മൾ പറയുന്നത്. അതിനകത്ത് വരുന്ന എക്സാമ്പിൾ ആണ് anthraquinone glycosides, and they will be showing 3 to 4 intense peaks between 220 and 290 nanometers. Clear? Now the second example is cyanocobalamin, that is vitamin B12, and it will be showing the maximum absorbance at 316 nanometer. Clear? So if any compound showing an absorbance at 316 nanometer, then you can assume that it may be a cyanocobalamin, and that's showing a maximum absorbance at 316 nanometer. Clear? So this is about the first application of UV spectroscopy, that is its use in the identification of plant constituent. So here the example is anthracnone glycosides, and they will be showing 3 to 4 intense peaks between 220 and 290 nanometer. Then cyanocobalamin, and that will be showing maximum absorbance at 316 nanometer. And the second application is, just 1 minute. Second application is used in the screening of plant extracts for particular class of compounds. Screening of plant extracts for particular class of compounds. So here the common example is, if a substance is showing a single absorption spectra between 250 and 260 nanometer. So if any substance is showing a single peak between 250 and 260 nanometer, then it may be, so it may be a simple phenol, then purine or pyrimidine or an aromatic amino acid. Screen. So it is used in the screening of plant extracts for particular class of compound. And here the common example is, if a substance is showing a single absorption spectra between 250 and 260 nanometer, then it may be a simple phenol, purine or pyrimidine or an aromatic amino acid. Clear? So these are the two common applications of UV spectroscopy. Then the third application is, it is used in the quantitative estimation of plant constituent. Percentage in the so it is used in the quantitative estimation of plant components. Clear? So these are the different applications of UV spectroscopy. So only three applications are there. One is identification of plant constituent. So here and example anthracnone glycosides, and they will be showing 3-4 peaks between 220 and 290 nanometers. And second thing is cyanocobalamin, that's vitamin B12, will be showing the maximum absorbance at 316 nanometer. And the second one is screening of plant extracts for particular class of components. And here, if a substance is showing a single absorption spectra between 250 to 260, then it may be a simple phenol, purine, pyrimidine or an aromatic amino acid. Clear? Now the third one is, it's used for the quantitative estimation of phytoconstituents. Clear? So that is about UV spectroscopy. Clear? Now we can see about the second spectroscopy, that is visible spectroscopy. So visible spectroscopy. So the working principle of visible spectroscopy is also same as that of UV spectroscopy. So here we already studied about UV spectroscopy, and regarding visible spectroscopy, the working principle is same as that of UV spectroscopy. But this spectroscopy is used in the estimation of colored compounds. Colored compounds ആണെങ്കിൽ അതിൽ UV spectroscopy ചെയ്യാൻ പറ്റില്ല. So there only visible spectroscopy can be applied. So visible spectroscopy is used in the estimation of colored compounds, and the wave range of visible spectroscopy is from 400 to 800 nanometer. So roughly നമ്മൾ പറഞ്ഞു UV എന്ന് പറഞ്ഞാൽ 200 to 400 nanometer ആയിരുന്നു. But roughly the wave range of visible spectroscopy is from 400 to 800 nanometer. Clear? Now we can see about the various applications of visible spectroscopy. So first application is, it is used in the identification of phytoconstituents. So here also it can be used in the identification of phytoconstituents. And here the common example is digitoxin. The lambda max is 495 nanometer. Then ergometrin, the lambda max is 545 nanometer. Then ergotamine, it is 578 nanometer. So this is the first application, that is, it is used in the estimation of phytoconstituent. Now second application is, it is used in the structural elucidation of phytoconstituent along with other spectral data. So ഇവിടെ ഒരിക്കലും നിങ്ങൾ ഇത് മാത്രമായിട്ട് structural elucidation പറ്റുമെന്ന് പറയില്ല. So it can be used in the structural elucidation of phytoconstituents along with other spectral data. Structural elucidation എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ determination of structure of a compound അത്രേ ഉള്ളൂ. So this visible spectroscopy is used in the structural elucidation of phytoconstituents along with other spectral details or data. Now the third application is, it is used in the quantitative as well as qualitative estimation of natural components. So or comparing it with authentic standard, etc. So it is used in the quantitative estimation of natural components by comparing with the authentic standard, etc. So these are the various examples of applications of visible spectroscopy. Clear? So the working principle of visible spectroscopy is also same as that of UV spectroscopy, but here only colored compounds can be estimated, and rough wave range of UV visible spectroscopy is from 400 to 800 nanometer. Principle എങ്ങനെയാണ് നമ്മൾ UV യിൽ പറഞ്ഞ കാര്യം തന്നെയാണ്. Means this light should be passed through a colored compound, then absorption absorption will be taking place. That time measure the intensity of absorptions as well as the lambda max. That's all. Then these are the applications. Okay. You can see about the next spectroscopy, that is IR spectroscopy. So IR spectroscopy is a infrared spectroscopy. It is a infrared spectroscopy, and the instrument used in IR spectroscopy is known as IR spectrophotometer. Ella spectrophotometer ennaana. So here the instrument used is IR spectrophotometer. Clear? So here the sample can be measured in solid or in the liquid form. So in IR spectroscopy, the sample can be measured either in solid or in the liquid form. And if it is in the solids, means in the liquid medium, then prepare a 1 to 5% solution with the help of either chloroform, carbon tetrachloride, or by the help of newjol oil or oil. So solution you have to prepare a suspension of means solution of 1 to 5% with the help of chloroform, carbon tetrachloride, or with newjol oil. Clear? And if it is in the solid form, then mix the sample with potassium bromide in the ratio 1:10 or 1:100. Then you have to prepare an anhydrous thin disc with the help of a mold and press. Then this disc can be used for the IR spectroscopy. Clear? And last thing is the range of measurement in IR spectroscopy is from 4000 to 667 centimeter inverse. Okay. So range of measurement is from 4000 or 4000 to 667 centimeter inverse, and it will be taking nearly 3 minutes for recording the entire spectrum. Only 3 minutes is required for recording the entire spectrum as per IR spectroscopy. Clear? So IR spectroscopy is the infrared spectroscopy, and the instrument used here is infrared spectrophotometer, and here the sample can be measured in solid or in the liquid form. And if it is in the solution, then prepare a 1 to 5% suspension with the help of chloroform, carbon tetrachloride, or with newjol oil. And in the solid state, mix the sample with potassium bromide in ratio 1:10 or 1:100. Then prepare an anhydrous thin disc with the help of a mold and press. Then the range of measurement is from 4000 to 667 centimeter inverse. Then it will be taking nearly 3 minutes to recording the entire spectrum. Clear? Now, ini idinte working principle endaanennulla karyam nokkam. So IR spectroscopy is concerned with the study of absorption of infrared radiation. So when a substance absorbs this infrared radiation, it may result in vibrational transition. So IR spectroscopy is concerned with the study of absorption of IR radiation, that is infrared radiation, and that result in vibrational transition. And here in a molecule, the atoms or group of atoms are connected by bonds. So that you already knowing that whether it is a means whatever molecule it is having lot of atoms, and these atoms or group of atoms are connected together by bonds, and in fact these bonds are not rigid in nature. They are not rigid in nature, so they will be having a natural frequency of vibration. Clear? So we are studying about the principle of IR spectroscopy. So it is concerned with the study of absorption of infrared radiation, and that result in vibrational transition. So in a molecule, the atoms or group of atoms are connected by bonds. Okay, and these bonds are not rigid in nature, so they will be having a natural frequency of vibration. Clear? And here, if you are applying an IR radiation, that is, if any IR radiation is passed through the sample, then it will reach a state. So when we are passing IR radiation through the sample, so we already studied that the sample will be having some natural frequency of vibration because of the bonds present along inside the molecule. Okay. So if the IR radiation is passed through the sample, it will reach a state at which the applied IR frequency equals to the natural frequency of vibration. So this natural frequency means the molecule will be having some sort of vibration, and that is known as the natural frequency of vibration. And if we are applying IR through this sample, then after some times a state will reach, and here the natural frequency of vibration equals to the applied IR frequency. Clear? And at that particular wave number, it shows a peak, and this is a IR spectrum. And this is a working principle of IR spectroscopy. Very simple. Okay. So it is concerned with the study of absorption of infrared radiation, and that result in vibrational transition. So in a molecule, the atoms or group of atoms are connected by bonds, and they will be having natural frequency of vibration because these atoms, these bonds were not rigid in nature, and they will be having some sort of vibration. Okay, so that's a natural frequency of vibration. So here, if an IR radiation is passed through the sample, it will reach a state at which the applied IR frequency equals to the natural frequency of vibration. And at that particular wave number, it will be showing a peak, and this is the IR spectrum. Clear? Now, so the region in the IR spectrum. So different regions are there in IR spectrum, near, far, അങ്ങനെ അതൊക്കെ നിങ്ങൾ അനാലിസിസിൽ ഡീറ്റെയിൽ ആയിട്ട് പഠിക്കും. നിയർ IR, far IR എന്നൊക്കെ പറഞ്ഞവിടെ പഠിക്കുക. So the region in the IR spectrum, which is about 1200 centimeter inverse, will be showing the band or peak, and that is due to the vibration of individual bonds or the functional groups. ഈ മോളിക്യൂൾ ഒരുമിച്ച് വൈബ്രേറ്റ് ചെയ്യുന്നത് കൊണ്ടല്ല. സോ ഈ റീജിയനിൽ ആ പീക്ക് കാണിക്കുന്നത്, അതായത് 1200 സെന്റിമീറ്ററിന് മുകളിലുള്ള റീജിയനിലുള്ള പീക്ക് കാണിക്കുന്നത് എന്തുകൊണ്ടാണ്? സോ ഇറ്റ് ഈസ് ഡ്യൂ ടു ദ വൈബ്രേഷൻ ഓഫ് ഇൻഡിവിജ്വൽ ബോണ്ട്സ് ഓർ ഫങ്ഷണൽ ഗ്രൂപ്സ്. ക്ലിയർ? And the region below 1200 centimeter inverse will be showing the peak due to the vibration of the entire molecule. So the region above 1200 centimeter inverse will be showing peaks or band due to the vibration of individual bonds or functional molecules, functional groups, and the region below 1200 centimeter inverse will be showing the peak due to the vibration of the entire molecule or the whole molecule. Clear? And this region is more complex. That is 1200 centimeter inverse means താഴെയുള്ള region is more complex, and this is known as a fingerprinting region. So the region below 1200 centimeter inverse is more complex, and this is known as a fingerprinting region. Clear? Since with IR spectroscopy, the functional groups can be identified. Clear? So ഒരു സ്ട്രക്ചർ നമുക്ക് കണ്ടുപിടിക്കണമെന്നുണ്ടെങ്കിൽ അതിൽ ഏറ്റവും ഇമ്പോർട്ടൻ്റ് ആയിട്ട് വേണ്ടതാണ് അതിന്റെ ഫങ്ഷണൽ ഗ്രൂപ്പ്സ് എന്ന് പറയുന്നത്. And this functional groups can be identified clearly with the help of IR spectroscopy. So we can say that this is most simplest as well as a reliable method in the identification of very different class of compounds. Clear? So simplest എന്ന് പറഞ്ഞാൽ it will be taking only 3 minutes for recording the spectra. Clear? So since with IR spectroscopy the functional groups can be identified, and hence it became the most simple as well as a reliable method of identifying the class of compound. Clear? Now we can see about the various applications of IR spectroscopy. So the first application is it is used in the identification of functional groups present in a compound. So the major application of IR is it is used in the identification of functional groups present in a compound. So here the example is alcohols and phenols will be showing peaks or band between 3600 and 2400 centimeter inverse. Clear? Now the second example under this category is aldehydes and ketones will be showing bands or peaks at 2750 centimeter inverse. And the third thing is esters will be showing peak or band between 1820 to 1680 centimeter inverse. And the next one is carboxylic acids will be showing peaks or bands between at 3520 centimeter inverse. Clear? So this is the first application, that is it is used in the identification of functional groups present in a compound. Clear? So adine nammal 3-4 example paranju, alcohols, phenols, then aldehyde and ketone, esters, carboxylic acid, etc. So their wave number or band is also already mentioned. So now the second application of IR spectroscopy is it is used as a fingerprinting device for comparing the natural products with their authentic standards. Natural products in their fingerprint kittum, adine nammal endu cheyum authentic standards ആയിട്ട് കമ്പയർ ചെയ്തിട്ട് അത് പ്യൂർ ആണോ ഇല്ലയോ എന്നുള്ള കാര്യം ഐഡന്റിഫൈ ചെയ്യാൻ പറ്റും. So this is the second application, that is it is used as a fingerprinting device for comparing the natural products with their authentic standards. Clear? Now the third application is it is used in the structural elucidation of phytoconstituents with other spectral data. So ഈ ഒരു ആപ്ലിക്കേഷൻ നിങ്ങൾക്ക് എല്ലാ സ്പെക്ട്രോസ്കോപ്പിയിലും എഴുതി വെക്കാം. Structural elucidation of phytoconstituents with other spectral data. Clear? So these are the different applications of IR spectroscopy. So yet another application is also there with IR spectroscopy, that is it's used for determination of the number of bonds, that is whether it is double bond, triple bond, etc., and the number of bonds, etc. So this can also determine with the help of IR spectroscopy. So these are the various applications of IR spectroscopy in the identification of functional groups present in a compound, then fingerprinting device, structural elucidation, as well as determination of number of bonds. So that is about IR spectroscopy. Principle എന്തായിരുന്നു? So means natural frequency of vibration is there. അതെന്തുകൊണ്ടാണ്? Because in a molecule means atoms are held together by bond and these bonds are not rigid in nature. It will be showing a natural frequency of vibration. So here if you are applying an IR radiation, after some times a stage will be reaching where both the IR radiation as well as natural frequency of vibrations are equal. Clear? Here it will be showing a peak and this is the IR spectrum. And this is the working principle of IR spectroscopy. So these are the different applications of IR spectroscopy. So that is about IR spectroscopy. So today we already studied about UV spectroscopy, visible spectroscopy and IR spectroscopy. And the remaining things you can see in the next video. So for the time being, thank you. Okay. Thank you.